Hello, welcome to the final review lecture for BE 209. This will be your best preparation for the final. The final is cumulative. It will include items from parts 1 and 2 of the course, but with a heavy emphasis on part 3. Because you already have review lectures for part 1 and 2, this review lecture covers only the material in part 3. A major theme for this portion of the course was the study of these three biopolymers. Following the central dogma, DNA makes RNA makes protein, the three different biopolymers, DNA, RNA, and proteins, and the methods used to interrogate them, DNA sequencing, microarrays, and mass spectrometry. In addition to how you study and quantify them, we talked about how you might manipulate them or use them to engineer systems to desired properties. The first lecture on this portion of the course was genome engineering and how we can manipulate DNA. The fundamental breakthrough which happened in the early 70s was the discovery of restriction enzymes which cut DNA at well-defined sequences. This ushered in the field of recombinant DNA technology, and along with PCR, these technologies ushered in modern biotechnology. Here we see several recognition sites for various restriction enzymes. We notice that the restriction sites tend to be palindromic, that they can be 4 bases, 6 bases, 8 bases, and other bases as well. Restriction enzymes that recognize 4 base pair sequences will cleave more often than a restriction enzyme that cuts with an 8 base recognition sequence. Restriction enzymes can produce blunt ends, in which case any pair of DNA molecules can be matched up with any other, or they can produce sticky overhangs, which is useful for assembling pieces based on sequence overlap, and which greatly increases the efficiency of the ligation process. One of the things restriction enzymes enabled was the production of molecularly defined DNA. In this case, we have a viral genome, which we cut with 2 different restriction enzymes, and it cut the genome a set number of times, producing bands of different sizes, but importantly, each of a defined sequence. Researchers at far locations can use the same restriction enzyme to cut the same genome, and they will have the same pieces of DNA available to work with them. These pieces of DNA that result from the restriction enzyme digest of the genome can be separated using a technique called Gel Electrophoresis. DNA is a polyanion, negatively charged. It migrates towards the positive end of an electrode. Smaller pieces migrate faster in agarose gels as they move more quickly through the obstacle course that's presented by the agarose polymer. Once a gel has been run, the DNA pieces can be visualized using a fluorescent dye called ethidium bromide, which interacts with the DNA, and it fluoresces in the visible wavelength when illuminated by UV light. That produces these pink orangish bands that we can see here. Another method, which is used less often, because if the DNA is labeled radioactively, then ordinary photographic film create an image of the emitted photons from the radioactive decay. But the important point is we can use a restriction enzyme to create specific molecules of DNA. We can use gel electrophoresis to separate them from each other and isolate a pure piece of DNA of defined sequence. Building on gel electrophoresis, we're able to identify specific pieces of DNA using the technique of southern blots. The separated DNA, according to size, is transferred to a plastic membrane, which by capillary action gets transferred to a plastic sheet that is next to the gel. This sheet is positively charged, nitrocellulose. It binds DNA tightly, and on this plastic sheet we have a duplicate of the electrophoretic gel. When we put this plastic membrane into a plastic bag along with a radioactive probe, then this probe can hybridize to its complementary sequence, allowing us to identify specific bands in the DNA. This technique of southern blotting can be used to genotype individuals. Here we have the example of the sickle cell gene. By using 2 probes, one for the normal gene, one for the disease gene, we can see that Person 1 only has the sequence for the wild type sequence. This is a healthy person. Individual number 2 only has the sequence for the mutant. This person has sickle cell disease. Person number 3 has both the wild type sequence and the disease sequence. This person is a carrier. This is done using southern blotting with 2 specific probes. Now, once we cut a piece of DNA with a restriction enzyme, the enzyme ligase has the ability to stitch it back together when the sticky ends or complementary ends can overlap or blunt ends are joined together. Ligase creates a covalent bond between the sugar phosphate backbone using a molecule of ATP for powering the reaction. This ability to ligate pieces of DNA helps us take PCR products, which are digested plasmids, which have 3 important features: a multiple cloning site, an origin of replication, which is not pictured, and a selectable marker, which is often an antibiotic resistance gene, allowing us to grow up this plasmid in the presence of antibiotic. In any case, both the plasmid and the PCR product are cut with the same restriction enzyme that gives them sticky overhangs of the same sequence. We're able to ligate in the piece of DNA, seal it up, and introduce the plasmid into a bacterium, put that bacterium into a culture which has antibiotic in it. Only those bacteria which have the plasmid can grow. We can isolate many copies of the plasmid, and if we've put in a gene, we might be able to use this construct to inside bacteria to make a large amount of a particular type of protein by driving expression of the mRNA from our plasmid that we've introduced into bacteria. One other very important biotechnological enzyme is the enzyme reverse transcriptase. This is the enzyme that broke the central dogma, which allows us to convert RNA into DNA. If we use an oligo DT primer, which binds to the poly-A tail of eukaryotic mRNA, we're able to create a strand of DNA that's complementary to the RNA. And then if we want to make double-stranded DNA, we can degrade the RNA and use remaining RNA fragments as primers to do second-strand synthesis. This double-stranded DNA copy of the original mRNA is referred to as complementary DNA or cDNA, and this is important for the study of eukaryotes because the mRNAs have the introns removed. We're not able to usually simply amplify a gene from a genome because the coding regions are separated by very large distances by introns. So in this way, using reverse transcriptase, we can make a DNA copy of a gene and use that in recombinant DNA. The other groundbreaking technique was PCR. PCR stands for Polymerase Chain Reaction. PCR is a 3-step process. The first step is heating it very close to boiling. This is in centigrade to dissociate the DNA into single-stranded DNA using thermal energy. The second step is the annealing of primers to the single-stranded DNA. These primers will allow us to create 2 new strands from this original piece of DNA, doubling the amount of double-stranded DNA. In the presence of excess primers, which are smaller and very abundant, will anneal more quickly than the 2 double strands reassociating over a longer period of time. And the third step is, once you have the primers bound to the DNA, then you're going to extend it, duplicating it. So the 3-step process is dissociation, binding of primers, and extension. By repeating those 3 cycles, you can double DNA each time. So with 3 cycles of DNA PCR, you have 8 molecules from your original double-stranded DNA. Like so many of the techniques we've discussed in this portion of the course, this technology and others depend on sequence-specific hybridization of complementary sequences. In this case, we've added 2 primers to amplify this specific section of DNA. We've already seen hybridization of probes to southern blots. We've seen hybridization of oligo-DT sequences to use reverse transcriptase. And of course, when we talk about microarrays, all these sequences will be hybridizing to sequences present on a chip. So the fundamental principle here is sequence-specific hybridization to complementary sequences. PCR, of course, can be used to amplify DNA isolated from cells And using reverse transcriptase, we can convert mRNA into DNA, and then we can amplify that DNA. So in essence, we're, by using the technique called RT-PCR, or reverse transcriptase PCR, we are creating copies of RNA using this DNA technology. In order to do PCR, you have to have the very fundamental skill of identifying PCR primers, and we've worked on that a little bit in the class. Most often people represent DNA sequences as a single nucleic acid sequence, and that's because the second sequence is inferred because it is completely defined. It's a reverse complement, meaning that 5' to 3', reverse, it's anti-parallel, and complement, because wherever a C is in the initial strand, a G is on the other strand. Wherever there's a T, there is an A. So when we amplify this piece of DNA, we have to pick two primers. One of them is going to amplify the top strand. One of them is going to amplify the bottom strand. The primers that you're going to have are going to be part of the initial sequence because you're duplicating it. So if you were picking primers and this was the sequence you'd have, and you would normally go longer than 6, you would say, okay, here is primer number 1. This is the region where I'm going to pick primer number 2, but I can't use this sequence directly, so I'm going to have to use G, A, G, C, and so on. And you know that that's the 5' end, so we're going to flip that around. This G is over here, and this AG is here. So you always write your primers from the 5' to 3' direction. This is a skill you need to have for the final. We also discussed a little bit why we might be able to make improvements in humans by doing engineering, even though evolution has had many millennia to achieve the present state of human evolution. And one of the main reasons is we have evolved under very different conditions. It's only in the last 100 years or so where the situations for reproductive success for humans has changed significantly. We no longer have great food scarcity. We have doctors to help us heal many illnesses which might have otherwise proved fatal and prevented reproduction. Physical strength is less of an asset than it used to be, and our hormonal chemistry and our lifestyles are quite different. And so accelerated engineering, accelerated evolution might be able to improve our fitness for our current situation. We also talked about Preimplantation Diagnosis. This is where DNA from a blastocyst, or at least an 8-cell fertilized egg, which would remove a single cell or the nucleic acid material in there, which we can amplify, we can sequence or genotype in a number of ways, and we can select particular fertilized eggs, which is currently limited to the avoidance of very severe disease. We next discussed in detail DNA sequencing. The goal of DNA sequencing is to find the order of A's, C's, G's, and T's in a DNA, and we did this primarily by using the technique of Sanger sequencing, also referred to as dideoxy sequencing, also referred to as chain-terminator sequencing. The cost of DNA sequencing has dropped exponentially in the last decade, and now we get to the point where full human genomes can be sequenced for a few $1,000s. It's very reasonable to expect this trend to continue, and so as James Galligan pointed out, it's useful to think about how you can configure different biological questions such that the readout will be sequencing DNA, as the cost will be soon next to nothing. This represents an overview of the entire DNA sequencing process. We have our original source DNA. It's fragmented into small pieces. Traditionally, using the sequencing technology of the 1990s, each of these sequences would be cloned. These plasmids with the various inserts are placed into bacteria. Each of these bacteria have a different insert. The plasmids are then purified, and we do the sequencing reaction. The sequencing reaction produces fragments of different sizes, and those different sizes are labeled with different colors, and this enables the readout of the sequence. The different fragments are separated with base pair resolution. Smallest fragments migrate the fastest, and we can see C, T, A, T, reading that out. We discussed the many steps needed to sequence a genome, and we also pointed out the steps which were very expensive and have been eliminated in the next generation sequencing or modern sequencing, which is responsible for the tremendous price drop which we just spoke about. The first step used to be fragmenting the DNA into lots of pieces, cloning them into different bacteria, producing a genomic library in which a genome is represented as small pieces in billions of bacteria. And then if we use one of the strategies called shotgun sequencing, we would take random clones, sequence them each, and then we have the process of trying to assemble them based on overlaps in the sequence. The fundamental molecular principle of the sequencing technique is the use of dideoxys, nucleotides. Here we have a normal deoxynucleotide that's used in DNA. It has a 3' hydroxyl. When it gets incorporated into a base, the strand can continue to be sequenced. When you put in a dideoxynucleotide, there's no hydroxyl here at the 3' position, and that will terminate the chain. You cannot synthesize that chain any further. Here we see how the 3' hydroxyl is involved in creating this phosphodiester backbone of sugar, phosphate group. When the hydroxyl is not present, the sequence terminates. So in every sequencing batch, there is a preponderance of normal nucleotides with 3' hydroxyl groups, a small percentage of dideoxys. They're mixed in together as we're sequencing a strand, or as we're synthesizing a strand more properly. Most of the time a normal base gets incorporated. Here we're focusing on the A's, and every once in a while a dideoxy is incorporated. Statistically, this can happen on any of the first 800 or so bases, and that will give us 800 different fragments, each differing by a base, which then we can read out on a gel. Here we show that by combining the 4 reactions into a single reaction and labeling them fluorescently, in this case A's are red, G's are yellow, we're going to produce fragments, each with different sizes and different colors, and these can then be read out on a gel. Looking at the sequence ATC, ATC, the color gives the identity of the nucleotide because the different bases have different color fluorescent molecules attached to it, and the length gives the nucleotide position. The smallest piece is indicative of the first base, the next one is the second base, and so on. If you have any confusion about this process and you haven't watched it yet, I highly recommend watching this sequencing video. Once we've sequenced individual clones, now we have to assemble them. The assembly is done by overlap sequences that will allow 2 different fragments to be put together. As we stitch the pieces of DNA together, we form contiguous pieces of DNA, which are referred to as contigs. The only problem with using overlap sequences for sequence assembly is the fact that our genome contains a great deal of repetitive DNA, and so that sometimes pieces that are not supposed to go together would be stitched together, and then we would miss out on information in between. This problem was solved by the use of paired end reads, meaning every time we clone a sequence, we're going to sequence both ends of the cloned fragment, and that we've made clones of different sizes, and so that whenever we're in a repetitive sequence, we have a chance that the other end is in non-repetitive DNA, and so when this moves over a little bit, we'll know that this end is moved over a little bit, and this will allow us to estimate the size of the repetitive fraction here. Now this is not 2,000 bases exactly, it's a distribution. 2,000 plus or minus 100 is probably an accurate representation of most of the fragments. For 150 kilobases, it's 150 kilobases plus or minus a kilobase. So we have to use a fair number of statistical tools to deconvolve the results, but it is possible and it has been demonstrated to work. The next generation methods eliminated a lot of the costly mistakes. One of the most important ones is the cloning of individual sequences into plasmids and bacteria, which is very laborious. In most next generation methods, either you're looking at a single molecule or you're taking fragments, you're ligating on primers that are universal to the same primers to each one of these sequences. Then in a single PCR reaction, we can amplify an entire library. By serial dilution, we can put molecules and capture them on a surface such that they're not too close or overlapping. We can use in situ amplification methods to make lots of copies of a molecule wherever one happens to land. And then we can do cyclic sequencing, where we're going to add different bases, different colored bases at different steps, and by recording the color each time a cycle is achieved, we're going to sequence each of these dots independently. The one I've just circled is a nucleic acid that has the sequence TAA. Another sequence here is ACG. When molecules are caught on the surface, if primers are also attached to the surface, the only way these molecules can be amplified is if the molecule bends over and hybridizes with a primer on the surface. This means that all duplicated molecules are isolated or physically located close to each other. This can also be achieved by putting beads inside oil-water emulsions, the PCR products that are occurring within a droplet, which have been diluted so that on average there's only one molecule with each bead in each water droplet. You're going to produce a bead covered with that PCR product, and then the beads can be put on a surface or captured in some way, and they can be sequenced individually. Once amplified, then we can do sequencing by incorporation of fluorescently colored bases. In this case, we use 100% fluorescently labeled 3' blocked nucleotides. We can, since it's 100% like this, at each location we're going to add a single base. We're then going to image it and record which color got incorporated in which physical location. Then we're going to treat it chemically. We're going to get rid of the fluorescent portion of the molecule. We're going to remove the protecting group of the 3' hydroxyl. This will allow us to do a second cycle where we incorporate another base, we image it a second time, and then we deprotect it a second time, and then we can keep going, adding a single fluorescently colored base each time. In this way, we can build up the sequence using multiple cycles. The next topic we covered was RNA analysis. The main motivation for RNA analysis is to answer the fundamental question about which genes get turned on in which cells under what conditions. All cells have a essentially identical genome. They have all the same number of genes, but by turning different genes on, they can express their unique phenotype. We mentioned 4 different ways of analyzing RNA, which we'll go through real quickly, but we spent most of the time talking about microarrays, which is the global method which enables us to study all RNAs of a particular cell at a single time. Northern blots are essentially like southern blots, except that we're loading RNA, separating RNA samples. RNA gets on the membrane, and we have a DNA probe, which can be, again, radioactively labeled or chemiluminescent labeled, and it can hybridize wherever it is on the plastic membrane, and therefore we can see which lanes contain that piece of RNA. And a type of experiment that is frequently done is in different lanes. We run RNA from different tissues, and we can see whether the RNA is present or not, or by using intensities we can get ideas about relative amounts of expression. In situ hybridization uses a probe that will hybridize to RNAs wherever they're produced. The probes are complementary to exon boundaries. We have exon number 1, exon number 2, which when the introns get removed, get stitched together. The probe sequences cross one of these boundaries so that it's not going to bind genomic DNA nearly as strongly as it will bind mRNA. To the probes, there are either fluorescent molecules or enzymes which produce colors wherever the probe sequence is retained. Here, the darker blue image indicates cells where the gene is being expressed. RT-PCR is the process of PCR amplification based on reverse transcription. We have an mRNA. We're going to reverse transcribe the RNA into a single-stranded DNA. We will degrade the RNA entirely. we will put in gene-specific primers and amplify that sequence of which we're interested in quantitating, and then we can run it out on a gel. The PCR products, in here we see 3 lanes in which we have high expression and 2 lanes in which there's low expression of a particular gene based on sequence-specific PCR. But the main method studied was microarrays, and it complicates our lives a little bit that there are 2 main formats of microarrays. One is spotted microarrays, and the other type is called Affymetrix GeneChips. In both cases, we're comparing 2 samples. In spotted microarrays, we take the RNA from one cell, the RNA from a second cell. We're going to label it with different colors, green or red. We'll mix them together. We'll hybridize them to a chip, and we'll record the relative amounts of fluorescence at each particular location. With Affymetrix chips, we're going to, again, compare 2 samples, but we'll do it on 2 chips. Each of them will be a single color. We'll take the RNA, we'll convert it into cDNA, we'll let it hybridize with a chip, we'll incorporate a fluorescent molecule on it, and we'll see where the RNAs will hybridize. Because of the high quality of each produced chip, we can compare 2 chips very reliably. On the spotted microarrays, each chip is a PCR. Each spot is a PCR product of the gene being amplified. For each of these spots on the chip, we know the sequence associated, so we know which gene is represented on which location, and so that if we see fluorescence at a particular spot, we'll know that that RNA was present in the cell under that condition. And for spotted microarrays, we get 4 different colors on the chip. We have red when the RNA is present in greater abundance in sample 1 rather than sample 2. They are green when the converse is true. The RNA is present more often in sample 2 than sample 1. We can get yellow spots when they're both equally present in both samples and at high levels. And then there are black spots, in which case the RNAs are essentially absent from both samples, and so there's no great amount of fluorescence produced at all. On Affymetrix chips, rather than spotting PCR products, we use the tool of photolithography to build up nucleotides one at a time, producing short oligos, 20 to 25 nucleotides. Initially, we start with a grid which contains millions of features. We have a photomask which allows light to only hit certain of those features that will deprotect those features, which will allow a nucleotide to come in and react with all the locations. So in this quadrant here, the base A will be represented in the first position. And this can be done with multiple cycles, not always, of course, being a fragment, but they can be essentially a checkerboard pattern where each position in the grid is addressed independently, and so that you can build up a sequence using multiple cycles of synthesis. Again, here you can see the 4 A bases, the 4 T bases that occurred after these 2 steps, but then after that the next base is ATCG, ATGC, and so each sequence is unique, each grid position is unique. Since these oligos are only 20 to 25 base pairs each, which is limited by the fidelity of the synthesis technique, we cannot cover the entire 5' to 3' region of a gene. We're going to have multiple probes per gene, in this case maybe 15 to 20, and they're going to be clustered near the 3' end, because we're always going to be sequencing using an oligo-DT primer starting at the 3' end. So this is the area where the cDNA is made most reliably. Furthermore, for each of these locations, we're going to produce a second feature, which is a mismatch feature, meaning that the perfect match is going to have the exact match to the gene. And then we're also going to put in a mutation, and this means the actual sequence will bind much less tightly, and so that the mismatch space is a measure of nonspecific binding, and this signal is subtracted from the perfect match to get the amount of fluorescence that is due specifically to the amount of RNA present in the sample. So here we see the overview of the experiment. We're going to extract our mRNA from a sample. We're going to label it, which means incorporating fluorescent molecules into the sequence. We're going to allow it to hybridize with the chip, where it's going to find its complementary sequence. That means it's bringing the fluorescent molecule along with it to the place where it's hybridizing, and then we have to measure the amount of fluorescence for each spot. The labeling reaction, which we'll just show here for the spotted microarrays, is that we're going to do reverse transcription, but we're going to incorporate amino allyl so that as we're making the cDNA molecule, we're putting in this extra chemical reactive group. Then we can chemically cross-link fluorescent molecules to this amino allyl group, and we'll make a fluorescently labeled cDNA, which can then hybridize with a chip. Once we have hybridized with a chip, we get an Excel spreadsheet which records the XY position and an amount of fluorescence. We can do that over and over again for many samples, for many thousands of genes. In this case, 1,500 are shown. And then the question becomes, what does all that mean? It's challenging for humans to interpret that much data. And so one of the first steps done analytically to analyze the data is using a technique of clustering, in which we're going to try to identify samples that behave similarly, and we're going to try to identify genes that behave similarly over a large number of conditions. And we hope that this will provide some insight into what's going on. Once we cluster genes and we get this kind of result, we'll make the hypothesis these genes are working on the same function. And if we know what the function of one of those genes are, we have a putative function for the maybe 50 to 100 genes that are pictured here. Similarly, if we find a number of samples that are all behaving similarly, then we'll have a hypothesis that these samples are in some way related. If we're looking at different diseases, we might say that these different samples have a similar disease. The way that those correlograms are produced is by calculating correlation coefficients between any two genes over the conditions. Genes which are highly correlated will show similar patterns of fluctuation, in this case high expression in condition 0 or 1, low expression in condition 2. This would have a coefficient of correlation of 1. Anti-correlated genes, where they're doing exactly the opposite, have a correlation coefficient of -1. No correlation is present if the correlation coefficient is 0. Again, since what we're going to try to do is find genes that are behaving the same way, we're going to look for genes that have high correlation coefficients. And the way they did that is we first had 5 different genes. We calculated the pairwise correlation coefficient of those 5. In the example, A and B had the highest correlation coefficient. We create a new entity called AB. Now we have 1, 2, 3, 4 items left. We recalculate the correlation coefficient of those 4 items, and we will combine those two that have the highest correlation coefficient. In this manner, we build up the connectivity, swapping rows so that the closest pair is always closest to each other. Here we compare the hypothetical example with a real example in which different genes have been put next to each other based on the similarity of their expression. Once we've identified a set of genes that behave similarly under a number of conditions, if we know what the function of one of those is, great, we can infer function for the rest of them. If we don't recognize any of the genes, we use a technique called enrichment analysis, which we'll use. There are many online tools which will enable us to use this type of analysis. Here's one. It's going to use the hypergeometric distribution to identify for a set of input genes based on the background of the total genome, are there any functions that are found more frequently than expected by statistical chance? If so, that constitutes an enrichment of that function and leads us to a hypothesis on the nature of why these genes are being clustered together, why they're behaving together in a biological sample. So to conclude this portion is we created DNA chips which had features for each gene in the genome. We took RNA, we labeled it, we hybridized it to the chip. We did multiple experiments on different conditions. We did hierarchical clustering to identify genes that behaved similarly, and then we did enrichment analysis to find what potential functions are of the genes which cluster together. Now, to intervene or engineer RNA level and produce different phenotypes of cells, we talked about two technologies: antisense oligonucleotides and RNA interference, or RNAi, which is going to help us manipulate transcript levels, which will then alter the amount of protein made, which will alter the phenotype of the cell. Antisense is a straightforward idea. You're going to include short oligonucleotides that are complementary to target mRNA, and this will interfere with the production of the protein. You can use modified chemistry so that it's more stable. The result is based only on the hybridization of the antisense oligo to the mRNA, and this will not completely knock out expression but reduce it significantly. And the mechanism is translational arrest. Here we have an antisense oligo binding to an mRNA. Here's the ribosome coming along the mRNA trying to make a protein. Once it encounters the antisense oligo, it cannot proceed, it stops. The protein is incomplete, and we have reduced the expression of that protein. Now, antisense was very exciting because it allowed us to target any gene, in contrast to drugs that we make to target proteins, which can be very laborious. But people trying to perform antisense experiments one time got very unusual results, that they saw that they had a phenotypic effect whether they used antisense or sense oligonucleotides. The explanation was that in both cases there was some contamination of double-stranded RNA, and that's what was producing the results in both those cases, and the single-stranded RNAs were not. Double-stranded RNAs are the active molecule in RNAi. A long double-stranded RNA will be chopped into short double-stranded RNA pieces, 21 to 22 base pairs. In this case, it's labeled short interfering RNA. You don't need to know that, but the short interfering RNAs will be bound by a protein called RISC, which will discard one of the two RNA molecules, and then it will use this RNA that it has retained as a template for binding. Whichever mRNA it binds will be cleaved by the protein into small pieces. This is an enzymatic process which accounts for its efficiency. Once this RNA is degraded, this RISC complex is free to go ahead and bind other mRNA molecules, destroying them. A lot of the power of this technique was when people started studying a worm called C. elegans, which eats bacteria. Special bacteria were created such that each different strain of bacteria had a different plasmid in it, which would make double-stranded RNA, and you target which genes you wanted to be knocked down simply by feeding the worms different bacteria. Here we see C. elegans, which is expressing GFP. They ate a bacteria that contained a plasmid that made double-stranded RNA, the same sequence as GFP, and after the worms eat the bacteria, then double-stranded RNA gets made. Dicer and RISC combine to chop up all the GFP mRNA. No GFP protein gets made. Comparing the two options, antisense is single-stranded oligonucleotides, RNAi is double-stranded, large or small RNAs. This is a technique based only on hybridization. It doesn't depend on any proteins. It's not very efficient, but it'll work in all species because all you're doing is binding to the RNA. RNAi depends on the host organism having dicer and RISC minimally. The efficiency can be fantastic as you have an enzymatic process which can amplify each of the effects of each of the individual initial double-stranded RNAs that are put into, and so it's only certain species in which you can employ this technique. The next set of videos looked at mass spectrometry and the analysis of proteins. We commented some of the reasons for analyzing proteins is the close relationship of protein to phenotype. Here we see the central dogma: DNA makes RNA makes protein, but since proteins are the molecules, the effector molecules, the molecules that do things, the relationship here between phenotype and protein is much closer than it is with RNA or DNA, and perhaps we'll have a better chance of understanding how a particular phenotype arises from a set of proteins. Also, we drug proteins. It's the primary way that we intervene in cells. And finally, proteomics allows us to study post-translational modifications, which, for instance, in the case of phosphorylation, can either turn a protein on or off. So whether you have a phosphorylated protein or an unphosphorylated protein is a very important question, not easily answerable by studying the RNA of a cell. This is the overall flow of a proteomics experiment. We have a set of proteins which we've isolated and separated in some manner. They're digested into peptides. The peptides are put in a mass spectrometer. We have a two-step process of essentially weighing intact tryptic peptides and peptide fragments. Then we're going to analyze the raw data. Each experimental spectrum will be compared to theoretical spectra. Then we'll eventually get a list of proteins which we've identified, and it's up to us to try and make some biological sense of that, which is very challenging. This slide again highlights that it's peptides that are the observable. We're not observing proteins directly. We chop up the proteins into peptides. We put the peptides on a liquid chromatography column. The most frequently used is a reverse phase column. This is going to bind things based on their hydrophobicity by putting in a gradient of organic solvent, decreasing the amount of water. We're going to be able to mobilize peptides of varying hydrophobicity. Hopefully we'll get one into the mass spectrometer at a time. The peptides that are produced here is a protein sequence. The enzyme trypsin, for instance, will cleave after arginine R or lysine K, and so we're going to be producing peptides, in this case 4 amino acids. And since these are peptides produced by the enzyme trypsin, these are called tryptic peptides. Once we produce the peptides, we have to get them into the gas phase. We didn't talk about MALDI much, which was matrix-assisted laser desorption ionization, but instead we focused a fair bit on electrospray ionization. And here we see our liquid chromatography column, which is very small, and the liquid is flowing at a very slow flow rate. Peptides are present in a highly acidic medium. Everything's protonated. There's also an electric field placed across the mobile phase in the mass spectrometer. The peptides are eluted in fine droplets, which the liquid will evaporate. Eventually you'll have coulombic repulsion causing the droplets to fragment, and then basically we have very close to naked peptides which are in the gas phase and will be analyzed in the mass spectrometer. Again, the tryptic peptides are analyzed in a two-step process. First you have an MS1 scan, which is a record of all the ions coming off the column at a particular time. Then we will identify 5 large peaks and analyze each of them in turn individually. We will create electromagnetic fields such that only a single ion mass is stable inside the mass spectrometer. We will then do an MS2 scan on that peptide, and that, together with the MS1 information, will reveal the identity of the peptide if it is a peptide. The MS2 scan looks at fragments of a peptide. Here's the original peptide. Each time it's been fragmented in a different location. It's rather remarkable that peptides happen to preferentially fragment at the peptide bond. So if we look at the difference between two ions, the B3 ion and the B4 ion, the difference is an amino acid. And when we see this, this will represent the difference in mass due to this fourth amino acid. I can't stress strongly enough, however, that this is not the way the raw spectra are analyzed. I merely go through it to show the relationship between the appearance of the spectrum and the molecular species which give rise to the MS2 spectrum. And then we look through the data acquisition process. We had the different ions coming off the column, getting trapped in the ion trap, and then proceeding to the detector, producing an MS1 scan. We produced conditions such that only a single mass is stable. We bombard those molecules with a helium gas to fragment them. We create the B and Y ions, which are the tryptic peptides fragmented at different peptide bonds, put those into the detector. We have some species that will weigh only a very small amount. They'll be down here. This is where the B1 ion and the Y1 ion go. We also have large tryptic peptides. These are large fragments. These will be on this end of the spectrum. And although I say the word weigh, we're not looking at only the mass. We're looking at the mass to charge ratio, m over z, but the charge status of any individual peptide is very easy to decipher for single-digit charges. So once we have a raw spectrum, which is an intensity profile of different m over z fragments, we have to put a molecular interpretation on that so that we can analyze the different spectra. The way we do that is we're going to compare each experimental spectra, which have been produced by digesting proteins into peptides and then fragmenting the peptides inside the mass spectrometer. We're going to compare that with theoretical spectra, which does the same process but in silico. We're going to take protein sequences, we're going to compute different tryptic peptides, we're going to then in silico fragment between every single amino acid, we'll produce theoretical spectra, and then we're going to do cross correlations between experimental spectra and theoretical spectra. And whenever we find a good match that is such a good match that it's above a threshold level, we're going to say that we identified that peptide and that, for instance, this theoretical spectrum probably resulted from this peptide sequence. For getting quantitative information on proteins, rather than what we did with mRNA, which was to label mRNA species red or green in quantitate fluorescence, here we're going to put on different labels that are going to be either light or heavy, because what we're doing is essentially weighing these fragments, and then we're going to mix them together. When they come off the column at the same time, we'll be able to tell by the different weights which peptide came from which sample, and we'll be able to calculate a ratio, and here the ratio would be about between 2 to 1 and 3 to 1, probably about 2.5 to 1, with the protein being more abundant in the heavy sample rather than the light sample. Modifying an amino acid with a chemical species, a post-translational modification, obviously is going to change the weight of that, and that's the mechanism behind our ability to identify post-translational modifications. The amount of the shift between, say, a normal lysine and acetylated lysine depends on the mass that's added. In this case, the acetyl group is 42 Daltons. And we do the analysis with acetylation and without acetylation. If including an acetylated lysine helps us explain more of the data, we will conclude that the lysine is in fact acetylated. The last point that we went through was the comparison of mRNA abundances and protein abundances, and we analyzed the relationship quite carefully. We were concerned that although we can draw a line which seems to describe the data, we wondered whether protein and mRNA abundances were really correlated. The issue here is that very close to the origin we have the great preponderance of the spots, and they don't fit the line nearly as well as these outliers. And what we found was that correlation coefficients are dominated by these few points out that are large, rather than the many more points which are at low levels. And when we calculated the correlation coefficient going in this direction, starting at the least abundant, ending up at the most abundant, we saw essentially zero correlation. Here it would be 0. This would be essentially no correlation for, in this case, close to 90% of the proteins. It's only the last 10% which showed a significant correlation, which would erroneously lead us to believe that mRNA and protein is highly correlated, but for 85% of the genes that's not the case. The next topic covered was a survey of computational resources available online. We looked at each of these listed here: EntreGene for gathering gene information, PubMed for the medical literature, the Genome Browser to get chromosomal location and gene context. We have a lot of experience with BLAST to identify unknown sequences, gene ontology, a controlled description of protein function, and pathway analysis to identify gene function in the context of other proteins and complexes. Entree Gene is the primary NIH repository of gene information. We enter search terms such as gene symbols or names of genes or numerical identifiers. We get a long list of information as summarized here on the table of contents. Very useful is a brief summary, a paragraph of each protein, what's known about its function. We won't go through it here, but things that were very useful is there is gene-specific literature present on the page. Interacting proteins are listed. General gene information, which also has the function, is listed in a separate section. We have the reference sequences to links to the RNA and protein sequence, and then links to many other websites, all containing information about a specific gene. PubMed is the most reliable database for querying the biomedical literature. We enter in a few search term keywords. They can be author, they can be title, they can be journal, they can be date. Your publications are listed in the summary format. If we expand using this drop down to the abstract format, we will have links directly to the individual articles online if Boston University has access to them. This search demonstrates that Boolean operators are operative and that we can build complex queries easily using the advanced search function. The most reliable view of the genome is present at the University of California, Santa Cruz Genome Browser. A wide variety of search terms can be entered, and you'll get back information about the gene. Here's P53, different splice variants, different due to alternative splicing of copies of the gene. We know that it's on chromosome 17. The red mark is on the P arm, not the Q arm, so it's 17P. And the genes that flank P53 is WRAP53 and ATP1B2, it looks like. The gene, second gene, third gene. Here, this is the space between two genes. And we can zoom in or out on these views very simply. We can also control what we see. Each one of these rows are what are called different tracks. We have approximately 100 tracks, which are here down off the end of the slide. We can turn them on or off or show how we want them to be turned on by using each one of these drop-down modes. Your preferences are remembered, so it's worth doing it once after familiarizing yourself with the different tracks depict. Here we're seeing splice variants of the gene. Here we're seeing how conserved it is. Here we're seeing where single nucleotide polymorphisms are within the gene. We're frequently in biology confronted with the fact that we have a certain sequence and we're wondering if it's a gene or what it does. The easiest thing to do is blast the sequence. BLAST stands for the Basic Local Assignment Search Tool. Depending on whether you're entering a nucleotide sequence, you'd use BLAST-N. If you're putting in amino acid sequence for protein, you would use BLAST-P. And you can control which database you search again. By clicking on the BLAST button, we'll then get output, which will tell how well we match, give us some statistics, and give us the name of the match sequence, and then our unknown sequence is presumed to be the same thing if the match is very good. We next discussed function, which is the Gene Ontology Consortium has produced a controlled vocabulary for gene function annotation. There are three major categories of functional information: the molecular function, the biological process, and the cellular component. And this vocabulary is hierarchical, and we'll see an example soon. When we go back to the Entree gene for the telomerase entry, we see that under molecular function, telomeric DNA binding was highlighted. If we go to the version 1 of the Gene Ontology browser, we can see telomeric DNA binding is a subset of sequence-specific DNA binding, which is a subset of DNA binding, which is a subset of nucleic acid binding, and eventually even just a subset of binding in general. These are all molecular functions. If we go back a slide, we'll see the different biological processes this can happen is telomere maintenance, replicative senescence when the cell has decided not to elongate its telomeres anymore. And so the molecular function is combined with the biological process and the location, which is mostly going to be the nucleolus and the nucleus. Here's the nucleoplasm. So it's attempting to give a complete description of the protein based on its vocabulary. The browser has an expandable version of the vocabulary, and this can be expanded by clicking on the plus sign so you can see all the biological processes, and then you can also identify proteins that are participating in that aspect by clicking on the numbers here. Lastly, we showed that the PDB, or the Protein Data Bank, is the primary repository for crystal structure information, and that there are various simple ways of viewing crystallized molecule to get a better idea about the structure-function relationship. Now, in addition to online tools, we thought about a little bit about a computational problem, and that was predicting genes from raw DNA sequence. We talked about a number of steps that we could employ, which we'll show here. Again, the problem is if we have a raw DNA sequence, which would be this entire 2,000 base pairs, how do we identify the gene regions, or the exons, which we've highlighted in green here? The first thing you do is you're going to break down your sequence into fragments. Each of those will be run into BLAST. Anything that has homology or that matches over a relatively large number of base pairs, if it has homology with a gene, it is likely to be a gene. You can also look for DNA regions which are conserved in multiple species. This most often identifies exons, but will also identify important controlling regulatory elements in promoters. And so if you observe this pattern of matching, you've probably identified a gene. We also look for open reading frames, or ORFs. We mentioned how evolution has made sure that there are stop codons in each reading frame. There are 6 of them, 3 forward and 3 backwards, for each DNA sequence. When we talk about reading frames for the codons, it's which group of 3 nucleotides are we going to try to make an amino acid out of. We can take the first 3 amino acids. We could take nucleotides 2 through 4, or nucleotides 3 through 5 as the 3 together that will define an amino acid. We can do this for the strand that we can see, or we can do this for the strand that we don't see. And instead of going in this direction, go in this direction based on the other sequence. And when we do that, if we submit it to the translate function, we can see that most of the reading frames, 1, 2, 3, 4, 5, have numerous stop codons. Therefore, you could not make a large protein from that sequence. But if we look at frame 2, going forward, there are no stop codons. It only codes for amino acids. This is highly likely to be a gene, whether we previously know of a gene with this sequence or not. Again, that's just an interpretation of what the nucleotides are saying. Then lastly, we can look for punctuation sequences to help us identify where genes are present if they're transcription start sites, or transcription start sites here, translation start sites, translation stop sites. Other things that can occur are TATA boxes in the promoter regions, and nucleic acid punctuation that codes for splicing. If you see a preponderance of these types of elements in any particular close location, it's a good bet that you have identified a gene based on all this punctuation. In the next to last class session, we discussed cancer, and we organized our conversation based on the landmark paper, The Hallmarks of Cancer by Doug Hanahan and Robert Weinberg, who looked at lots of papers that described various aspects of the biology of tumors and synthesized all that they read into a recognition that there are 6 fundamental processes which cancer cells have altered relative to normal cells, which were self-sufficiency in growth signals, meaning that even if there's not a ligand binding that the cells will divide, it has to be insensitive to anti-growth signals, meaning tumor suppressors are not active. It has to be able to evade apoptosis, which is a normal cellular result in response to, for instance, DNA damage. Tumor cells will not commit apoptosis despite a lot of DNA damage. It has to have limitless replicative potential, meaning that the telomeres, which normally get shorter each cell division, will be elongated by telomerase. That you need to create angiogenesis so that you can have blood vessels that will support the growth of large tumors. And then tumors have to make places for themselves. They have to invade existing tissue and metastasize to other regions. We pointed out that by the time tumors are detectable, they've already been growing for many cell doublings, and things are already well on the way to having significant and bad disease, or even succumb to the disease. So that pushing detection early and earlier is one of the best things we can do to prevent serious consequences from the appearance of cancer. Cancers occur when we accumulate mutations. Mutations can come from the environment. They can come from specific molecules that will covalently modify DNA. It can come from ultraviolet radiation or X-rays, which can produce either thymidine dimers or double-strand breaks. It can come from a variety of molecules in the environment, one of the most important being all the free radicals that are present in every puff of tobacco smoke. The common denominator here is damage to the DNA, which will cause the acquisition of those 6 biological processes which cancer cells need in order to produce damaging disease. We talked about 2 fundamental classes of cancer-related genes, oncogenes, or proto-oncogenes at first. When they get mutated, they become oncogenes. These are like the gas pedal, the things that make cancers grow. And then the equivalent of the brake pedal are tumor suppressor genes. These are the genes that normally keep cell cycling in check. When they get mutated, the brakes no longer work and you will have cell cycling. Mutations in oncogenes or tumor suppressors are different quantitatively. Since oncogenes are like a gas pedal, if either one of your 2 copies gets mutated, all of a sudden you're pressing down on the gas, you're going to have cell cycling. These are gain-of-function mutations, meaning that the protein that's mutated is going to do something that it wouldn't normally do. Loss-of-function mutations occur in tumor suppressors. In this case, the tumor suppressor function, both of them need to be inactivated before you're going to have undesirable phenotypic consequences. If only one copy of a tumor suppressor gene is mutated, then you have the good second copy. The brakes are intact. You're not going to see a big phenotypic change. Because each mutation is a relatively rare event, for both copies to be inactivated, that would be extremely rare, and thankfully it doesn't happen that often. An example of an oncogene and a gain-of-function mutation was RAS. When it's mutated, it loses the ability to hydrolyze GTP. GTP remains bound to RAS. This leaves it stuck in the on position. It's going to activate downstream proteins, including the MAPK signaling pathway. That's going to lead to cell growth and cell division. This is inappropriate signaling because there's no ligand here binding to the receptor, which is telling it to be bound to GTP, which is the normal signal for downstream signaling. An example of a tumor suppressor is the retinoblastoma protein, RB, which normally binds to transcription factors, inactivating them, preventing cell cycle. When RB gets inactivated, whether by regulation or mutation, then it's no longer bound to the transcription factors, which will then go ahead and proceed to transcribe genes that are necessary for progression in the cell cycle. In addition to mutations, another type of DNA change that can occur that can lead to cancer is a chromosomal translocation. In a very famous case, the Philadelphia chromosome, because it was identified in Philadelphia, had a translocation between chromosomes 9 and chromosomes 22. This causes chronic myelogenous leukemia, or CML. And what's occurring in the translocation is a breakpoint which places a very strong promoter the BCR promoter, in front of a growth-promoting gene, ABL, which is a tyrosine kinase. Now, by making a great deal of this molecule, because it's under a very strong promoter, you're making much higher levels of mRNA, you're going to get much higher levels of protein, and this leads to cell cycling. That's inappropriate. This would be a protein you would want to inhibit with a drug to prevent the downstream signaling. Translocations are actually very common in cancer, and as we can see a cancerous karyotype to a normal karyotype, and here you see multiple abnormalities, multiple copies of genes, multiple translocations, two different colors. Cancers often produce this sort of tipping point phenomenon. Once DNA damage starts to accumulate, it has sort of a runaway function, and the genomes can get thrashed pretty significantly. Early studies in the 1990s by Bert Vogelstein and co-workers Doug Fearon examined many different colon cancers and noticed patterns in the progression of what could be seen under the microscope. By sequencing lots of genes, they were able to correlate various genes getting mutated with different appearances under the histology. And this was one of the first papers beyond the initial two-hit hypothesis to suggest that multiple genes needed to be mutated in order for a cancer to occur, which was an important precursor for the Hanahan-Weinberg paper. Again, I've gone through these 6 hallmarks of cancer, which you should be familiar with for the test. It's generally a good idea to know a protein that is involved with those functions, or at least what type of genes might be associated with that kind of function. We mentioned how the particular framework lends itself to making predictions about mutations and how we can anticipate whether a new mutation would be likely to cause cancer. The types of mutations that do cause cancer are ones that complete mutations in all 6 of these groups. And here we see 5 different ways of knocking out each of these biological processes. But we can see in some cases there are redundant mutations which might have slight quantitative effects but are not probably necessary to cause the cancer. To demonstrate this process, if we had a cell that had 4 mutations in it and we had to figure out which 2 mutations would be most likely to cause cancer, we would be sure to see that we have all 6 of the categories, and we see that this here is the mutation set that would complete that and produce the most aggressive phenotype. We mentioned that 85% of all cancers occur in epithelial cells originally. These are the cells that are the direct interaction between the body and the environment. They sit on top of a basement membrane, and when they start to proliferate, they will initially proliferate into the lumen or into space, but not invade the body. When these cancers eventually break through the basement membrane, this is called an invasive cancer. This is a cancer that's likely to metastasize, and unfortunately this would have a very much worse prognosis. For diseases like breast cancer or prostate cancer, there are a lot of ducts in which the ducts are considered essentially to be the outside. They're surrounded by basement membranes. Benign tumors will start to proliferate, can obliterate the lumen of these ducts, but eventually the right cocktail of extracellular matrix digestive proteins, proteases, collagenases will be produced in the tumors. They will cut through the basement membrane, and then they will lead to advanced disease. Once it breaks through the basement membrane, it will promote angiogenesis. That means it will recruit blood vessels to help nourish it. Various cancer cells will also be able to gain access to the bloodstream and use the bloodstream or the lymphatic system as a distribution channel to colonize other locations in the body. This is a metastasis. They will, for some reasons, stick to various places in the endothelium. If they're able to diapodese through the endothelial wall, they will gain access to new tissue areas. If they are able to establish a foothold in the new environment, they'll be able to proliferate and cause a metastatic lesion. And by the time tumors are detectable, they're typically shedding several million cells per day. And so even though this is a case where most of the cells will not successfully metastasize, since there's so many being released that metastases can form rather quickly when tumors are of macroscopic size. Tumors are capable of releasing molecules that will stimulate blood growth towards them. They also are able to hijack parts of the immune system, which will trick into helping them seduce blood vessels to grow towards them. And the greater the vascularity of a tumor by blood or lymph, the more likely that you're going to have metastatic disease. We discussed various high-throughput analysis of cancer. This was a technique called comparative genomic hybridization, in which we use DNA chips to, rather than hybridize against mRNA samples, we hybridize them against DNA samples. We saw that in cancer some regions are duplicated. That means they're present more than twice in the genome. This is where regions that are likely to harbor oncogenes. There are areas which are deleted. These are areas which are likely to possess tumor suppressor genes. And if you think back as to what a cancer karyotype looks like with all the various colors, you'll see how we get amplified or deleted regions. Of course, many cancers are getting sequenced, and sequencing is becoming so cheap. The pattern that is seen over and over again is that there are a relatively small number of genes that are mutated in a large percentage of a particular type of cancer, and what instead is seen is many genes that are mutated only rarely, but can each contribute to the cancer phenotype. When these genes are analyzed, one pattern that has emerged is that various pathways can be mutated frequently, even though the individual proteins are mutated more seldomly. We talked about microarrays used to classify leukemias, that if we have a large number of genes, we can have essentially a vote on whether a sample might be ALL or AML. ALL would have high expression for this list of genes and low expression for this set of genes, whereas for AML that situation would be reversed. We also saw in another example in breast cancer how microarrays could be used for prognosis and subtype discovery. Here we're simply using it to diagnose two different types of cancers. And this was the example of subtype discovery for breast cancers. And by subtypes we mean types of breast cancers that showed the same patterns but which were different from other types of breast cancers. And then we saw that these various subtypes had different prognoses in terms of expected cure rates or survival times. We concluded by a look at a way to engineer cancers from normal cells, and that was the incorporation of three proteins, RAS, telomerase, and this viral protein called SV40 large T antigen. This interferes with the normal functioning of proteins P53 and the retinoblastoma protein, so by making this one change, we're knocking out both of those genes. And the three genes and their accumulated impacts will modulate the biological processes that are required for cancerous growth. This is not something you would want to do necessarily. It's not treatment related, but it was an important conceptual advance in showing that particular genes, when mutated, will reliably lead to tumors. This figure was used to explore the six landmarks of cancer and also serves as a reminder of many of the topics which we've learned this semester. We concluded the course by a discussion of stem cells, starting with a highlight of the fact that the embryonic stem cells which are present in the inner cell mass of the blastocyst will eventually lead to all tissue types present in an adult organism, specifically different tissue types from the three major germ layers, the ectoderm, mesoderm, and endoderm, and also germ cells themselves. Not only are stem cells able to produce all these different terminally differentiated cell types, they're also capable of an asymmetric division in which, as a stem cell divides, one of the cells will become one of the differentiated cell types and the other cell remains a stem cell. This allows for self-renewal, and together these characterize stem cell capabilities, meaning that they're pluripotent and essentially immortal because of self-renewal. In terms of nomenclature, there's a difference between true fertilized eggs, which are totipotent and can give rise to an organism, versus embryonic stem cells, which are pluripotent, meaning they can form any cell of the body, but not an entire organism itself, because they are not capable of producing the structures of the placenta. We highlighted that there are different types of stem cells. There are the embryonic stem cells, which have the advantage that they can be turned into any particular cell type in principle, and that we can have an abundant supply of them because they propagate in vivo so well. Contrasted that to adult stem cells, which are generally tissue specific. They are not immortal as embryonic stem cells, although they have much greater replicative potential than normal cells. They are committed to a certain subset of cell types, and there might be a type of adult stem cell for just about every tissue type. There are induced pluripotent cells, which are created by taking an adult cell and de-differentiating it into an embryonic stem cell-like condition. And then there are also cancer stem cells, which might be a small percentage of any cancer, but which might go a long way to explaining why cancer treatments are often ineffective. We discussed a little about the controversy surrounding embryonic stem cells and their use, which are primarily controversial because it involves destruction of a fertilized egg, which in principle could go on to being a human being. Other issues are that they can be immunogenic or tumorigenic, meaning the immune system could reject an embryonic stem cell obtained from a different source, or that it could go on to cause cancers. These are generally not the case or the considerations for adult stem cells. However, it's hard to obtain them. They're in limited quantity because we can't propagate them indefinitely in vitro, and they also age. They don't have unlimited replicative potential. One of the amazing things about stem cells, which evolution has engineered, is that stem cells tend to selectively retain the original DNA strand. As we know, every time a DNA strand is copied, you're likely to introduce mutations. Because stem cells already have so many of the properties that cancers have, you don't want them accumulating too many more mutations. And so if you're always keeping the original strand with a stem cell, then you are less likely to accumulate mutations and tip the stem cell into being a cancer. I should say the mechanism by which it selectively retains the original DNA strand is not known. We talked about 3 different methods for producing pluripotent stem cell lines. One is from embryos that are part of attempts for in vitro fertilization. Once that process is concluded, we have the case where there are still fertilized embryos sitting in freezers, and if we harvest the inner cell mass, that can produce stem cells. You can do somatic cell nuclear transfer, SCNT, and we'll see that. And finally, you can produce induced pluripotent stem cells by inserting 4 genes and de-differentiating the cell type. So here we have the way that's involving blastocysts. This inner cell mass, which is present in day 5 after fertilization, contains a large number of embryonic stem cells. These cells can be isolated, put on in petri dishes on top of a second layer of cells, and with the right factors they can be maintained as embryonic stem cells. You can also do the somatic cell nuclear transfer. Rather than having 2 haploid genomes fused to produce a diploid genome and go on to be an organism, you can remove the haploid nucleus from an egg. You can take the diploid nucleus from a somatic cell. You can insert that very carefully into a gene. You activate it so that it starts cell division, and you can use this method for what's being referred to most recently as therapeutic cloning, meaning that the goal is to produce individual cell types or tissue types that can be useful in repair of diseased organs. Then lastly, the IPS method is there are a number of transcription factors. Here are 2 solutions to the problem, where if you add these transcription factors to a differentiated cell, you'll get an undifferentiated cell, which then exhibits the properties of pluripotency. Some of the concerns are that some of the genes are also known as oncogenes, and that it's dangerous to create cells by specifically incorporating these genes. And it is true that most attempts at stem cell therapies have produced elevated rates of cancer formation. So one thing that if people have been trying to do is to incorporate these gene products in ways that, well, they'll be inactivated after a short time after the cell is dedifferentiated. One way of doing that has been trying to add the proteins rather than the genes themselves. The proteins will eventually be degraded, and that one would hope that would prevent the formation of tumors. This is an area that's being actively explored. To keep embryonic stem cells pluripotent, there are a couple things that are needed. We need to put in a particular ligand called the leukemia inhibitory factor. The embryonic stem cells have to be sitting on a feeder layer. These are a layer of cells which are secreting some substances which we're not entirely sure of, but these cells plus LIF are required to maintain an embryonic-like state of the embryonic stem cells. We talked about the reverse problem is that once you've got the embryonic stem cell, how do you get that converted into tissues of interest? And we showed the disappointing initial studies where specific growth factors, which were thought to be fairly specific, were added to embryonic stem cells, and we found that most of these single item factors were not nearly as specific as we would like, which has led to the hypothesis that in order to develop a particular cell type, like for instance if you're interested in regenerating heart tissue, you need to have a series of growth factors in the right combination and in the right timing to recapitulate the developmental pathway of embryonic stem cells turning into heart tissue. We contrasted that to the better success of using an adult stem cell, which in this case is the hematopoietic stem cell, which gives rise to all the white blood cells and the red blood cells required in your circulation. The factors and genes which need to be turned on are much better understood in this case, and so while we have the disadvantages of a smaller supply and a limitation in terms of what types of cells they can turn into, we are able to manipulate this process by the selective activation of a small number of transcription factors, which will change the cell lineage development to these various different outcomes. And we concluded this with a discussion that stem cells, although filled with promise, have not led to a large number of therapeutic successes. We went into a little bit of detail of how stem cells are potentially going to help gene therapy, where we could correct mutant copies of genes. Standard gene therapy has attempted to incorporate corrected copies of genes into viruses, having these viruses infect target organs, allowing the replacement of the correct gene into places where a mutated gene is. This has led to a couple of problems. You have large immune responses to viral infections, and it's hard to get sufficient infectivity so that you have a large enough portion of the organ being infected and getting proper copies of the gene. The idea with stem cells is that if we could take fibroblasts, skin fibroblasts, convert them to iPS cells, incorporate a corrected copy of a particular gene into a single embryonic stem cell, then that could be propagated as long as we kept track of which cells had the corrected gene or not. We could put those stem cells back into the patient. There would be no immune response since the stem cell originated from the patient's own skin cell, and over time these cells would have plenty of time to repopulate a target organ and hopefully minimizing the impact of the mutated gene in the original tissue. Okay, that concludes this review. Good luck on the final exam. Regardless of your performance on the initial 2 exams, go into this exam confident that if you perform well, that your final grade will reflect the amount of knowledge that you now have. It's been a pleasure working with you this semester, and I wish you well in your future endeavors. Thank you for listening.